Hello, everyone, and thank you all for being part of the annual GRCF Symposium. My name is Melissa Olson, and I am the director of the GRCF. Although this year is virtual, we are very excited to bring you some amazing science in a new format for 2021. The GRCF has been a part of the Johns Hopkins community now for more than 30 years. And it's because of the support of researchers and colleagues in this audience that we continue to grow as a part of the JHU community. I'd like to take this opportunity to thank you for your continued support and trust in our facility. A few housekeeping announcements. This presentation has been pre-recorded. As such, Dr. Modell will be available throughout the course of the presentation from 1 to 1:55 p.m. to answer any questions you may have. Please feel free to post your questions to the Q&A board throughout the presentation. It's my great honor today to present to you Dr. Joshua Modell. Dr. Modell is an assistant professor in the Department of Molecular Biology and Genetics at Johns Hopkins University. Here he studies the interactions between bacteria, CRISPR-Cas systems, and bacteriophage using an integrated approach combining molecular biology, next-generation sequencing, functional genomics, biochemistry, bioinformatics, and time-lapse microscopy. His lab explores fundamental and mechanistic questions at the interface of CRISPR, bacterial, and bacteriophage biology. Dr. Modell has a long history in science. He studied sea urchin development at Duke University with Dr. David McClay. He worked for 2 years with Dr. Todd Golomb at the Broad Institute in Cancer Genomics. He studied cell division and bacterium with Dr. Michael Laub in his hometown of Cambridge, Massachusetts at MIT. Dr. Modell did his postdoctoral training in New York City at Rockefeller University, studying CRISPR-Cas biology with Dr. Luciano Marafini. In 2018, Dr. Modell joined the faculty at Johns Hopkins University, where he continues to study the natural biology of bacterial CRISPR-Cas systems. It's my pleasure today to introduce our keynote address speaker, Dr. Joshua Modell, and his seminar titled, A Natural Single Guide RNA Repurposes Cas9 to Autoregulate CRISPR-Cas Expression. Without further ado, Dr. Joshua Modell.
Hello, my name is Josh Modell. I'm an assistant professor in the Department of Molecular Biology and Genetics. It's a great privilege to give a seminar at the 2021 GRCF Virtual Symposium. I'm going to tell you about my work on CRISPR-Cas biology. Before that, I just want to say I've always been a big fan of NGS sequencing technology, and I know your lab is only as good as its sequencing core. And so when I got here in 2018, it was really important to me to establish a relationship with our GRCF core, and we've done some fantastic work with them, and them with us, I should say. And so I'm excited to tell you today about some of the work we do and some of the ways I think you'll see in which we use deep sequencing. So just to give you a quick overview, my lab, we do study CRISPR-Cas biology. We do study Cas9. But we are not a tools lab, so we don't develop Cas9 tools. We actually don't really do any editing either, although we hope to soon. But we study the basic biology of how these systems work in nature, which involves how bacteria use these systems as immune systems to defend themselves from foreign agents like shown here, the bacteriophage, which is infecting this bacterial cell. And so today I'm going to focus in particular on how these systems are regulated. Any organism with an immune system knows that it's important to have potent immunity against a foreign agent while also mitigating autoimmunity. And so we're going to talk today about how these CRISPR systems are regulated. In particular, I'll touch on our recent paper, came out a few months ago, demonstrating that Cas9, which until now we sort of thought of as this programmable nuclease, it turns out it's also evolved as a transcription factor. And in nature, it uses a very strange non-canonical guide RNA to target and repress its own promoter. So the regulation of this CRISPR system actually occurs using an intrinsic regulator, which also lives within the system. So first, to take a step back, we are a bacteriology lab. I probably don't need to convince you that we all live in a world of bacteria. So this is a swab of different strains you can find on a subway pole in New York City. We have lots of bacteria in our gut, all over our skin, and in the soil. And there's also many pathogenic bacteria that are increasingly becoming very problematic. So here's showing a tuberculosis patient that with diarrheal diseases, each kill about 1 million people a year worldwide. And there's a growing threat of antibiotic resistance bacteria. So here I'm showing Acinetobacter baumannii and Pseudomonas aeruginosa, which are two examples of strains that are increasingly difficult to treat with current antibiotics. So this is a huge issue. And I think the CDC estimates that by 2050, there might be 10 million deaths a year caused by antibiotic resistant strains. So this is really something that we need a lot of attention on. And I think what I'll show you today is there's something new on the horizon as an alternative to antibiotics for the treatment of bacterial infections, and that is bacteriophage. So as much as we live in a world of bacteria live in a world of bacteriophage. These are the most abundant organisms on the planet. So there's 10 to the 31 bacteriophages on Earth. And so for every bacterial cell, there's essentially 10 phages out there trying to get in and cause what you see here, which is a lytic event. So a single phage can infect a cell, and 30 minutes later, there's about 200 of them waiting to burst the cell and then infect its neighbors. And so just the amount of phage is staggering. If you consider all the grains of sand on Earth and you consider each grain as its own brain, then the number of neurons you would have on the planet is the number of phages. So this relationship between bacteria and phages, to me, is really fascinating. It's led to a constant arms race where each organism is trying to attack and defend itself from the other. And as a result, bacteria have evolved several different types of immune systems. They have an ability, some bacteria, to do what's called abortive infection. So this is a recognition that a cell is infected, and that cell will commit a sort of altruistic suicide to kill itself before allowing the phage to reproduce and lyse the cell. So while that cell dies, it's able to protect its neighbors because it doesn't release phage. Other cells have immune effectors called restriction enzymes. We're going to touch on these a bit later, but these are nucleases that are programmed to cut a specific sequence when it occurs in the phage, but not when it's within the host. These two systems are innate immune systems, and they're innate because they are sort of looking for the same features of a phage whenever that phage shows up. We also now know that bacteria have acquired immune systems, and that's what a CRISPR-Cas system is. And so when I say acquired, this means it can acquire memories of previous infections, and it can use those memories such that upon a subsequent infection, that foreign agent is killed much more quickly. And so you see in bacteria that there are layers of immunity. Just as we have innate and immune and acquired immune systems, so too do bacteria. And so I'll just briefly touch on, I think, what motivates our research into bacterial immune systems. One is that what drew me to the field is this fascinating idea that in a single bacterial cell, you can encode all the features of an acquired immune system, which in us takes, a dozen different cell types and billions of cells, each talking to each other in different ways. Somehow this happens in a single bacterial cell. And so we're really still trying to understand a lot of the basics of how this works. And I find that super fascinating. Two is that, as I mentioned, this arms race between these two organisms has led to many steps in evolution in these bacterial systems. And so phages, because they are so potent, bacteria have evolved defense systems. They've evolved proteins to sort of evade detection by these phages. But bacteria have also acquired genes from these phages. So horizontal gene transfer is a mechanism of picking up genes from neighboring cells. And in fact, bacteria can acquire genes from a phage. If they can survive the infection, they can pick up genes. And a lot of the pathogens that I showed on the first slide, they're pathogenic because of toxins, which were actually acquired, many of them on prophage genomes. Those are when phages become dormant. They become prophages, but their genes become accessible to the bacterial host. With both restriction enzymes and CRISPR systems, we've once we understand their mechanism, been able to develop these incredible precision tools, which with restriction enzymes led to a revolution in molecular biology. And now with CRISPR systems, there's been a revolution in gene editing. So as we understand these systems better and better, there's always this potential to make new and better tools. And finally, as I mentioned, there are new opportunities now for phage therapy. If you live in Russia or in the country Georgia, you may have heard of this, but in the West, we sort of rely on antibiotics. But there's many anecdotal cases now where phage therapy has helped patients with really recalcitrant infections. And so I think increasingly you're going to see a lot of work done in this arena, especially as antibiotic resistance sort of takes over. So now I'm just going to give a brief history of bacterial immune systems. Work done in the early to mid 20th century by Salvador Luria, there was initial observation that different bacterial strains, which were otherwise very similar, could differ in how they respond to a phage infection. So some cells would lyse, while other cells seemed to mysteriously be somehow protected. And years later, Werner Aber, when he looked inside these cells that were protected, he noticed that the DNA from these foreign agents was actually being chopped up. So this led to this first idea that there was some sort of system within these resistant hosts that was cutting phage DNA while sparing the host. And then finally, work done by Hamilton, Smith, and Daniel Nathans here at Johns Hopkins, actually in my own department, discovered that this immunity was provided by restriction enzymes. And so a restriction modification system is programmed to cut a sequence of DNA. Here's the classic EcoR1, which cuts GAA-TTC, but it only cuts that sequence if it's unmethylated. And so the key here is there's another protein called a modification protein, which will methylate that exact same sequence. And so in the host, this sequence is methylated and protected, but when a phage injects its genome, it's unmethylated and therefore will be cut. So Dr. Smith and Nathans were awarded the Nobel Prize for, along with Werner Aber, for these discoveries on restriction enzymes. Of course, molecular biology was enabled by these discoveries. But you might ask a question, why didn't they win the Nobel for CRISPR-Cas systems? Why didn't we find these other defense mechanisms and back in the 50s, 60s, and 70s? And you might ask, did they just simply choose the wrong model system? So CRISPR-Cas systems are found in only 50% or so of bacteria. And if you look at the E. coli strains that they were using, which were sort of the workhorses of bacteriology and phage biology, it turns out they have beautiful CRISPR systems. So they're fully functional, they're intact, but they are not active against phage. And this is because they are repressed by this host factor called HNS. And the more we've learned about CRISPR systems since, the more we're seeing this trend where they seem to be constitutively either silenced or repressed such that they're only made at very low levels. And as you'll see, we think this is because CRISPR systems, while they are potent against foreign agents, you also have this issue of autoimmunity. And for that reason, you should keep the system lowly expressed. And so then how you turn it on is a really interesting question that we're still working on. So there's lots of different types of CRISPR systems out there. I'm going to focus today on the system we study in my lab, which is the type 2 system from Strep pyogenes. This includes the Cas9 that everybody knows and loves in the tools and editing world. But there's lots of different systems out there. And if you're interested, I encourage you to kind of take a look at the different subtypes. There's 6 major subtypes. They each have different activities. Some target DNA, some target RNA, some cut DNA, some cut RNA. So there's a lot of really interesting mechanisms and tools that are being built from each of these systems. But we study type 2 systems. And while it's known that the type 1 system in E. coli is repressed by HNS, we don't know more broadly how CRISPR systems are not only turned off, but dynamically regulated. So we wanted to know in our system, one, what regulates the system, and secondly, how is the system turned on when it's needed, for example, during a phage infection. And I don't know if I'm blocking the screen here, but there should be a phage you see up in the upper right. So these are the questions we wanted to go after. And so now I'm just going to give you another brief history of CRISPR-Cas systems. And in doing so, just try to explain a little bit about how they work in nature, because for the talk, you'll need to know a little bit about these mechanisms. So CRISPR systems were first observed in the 80s. There was a Japanese group studying alkaline phosphatase, which has nothing to do with CRISPR biology, but they just noticed that the DNA downstream from their genes of interest looked kind of interesting. And so this is the first publication with a CRISPR locus. If you look closely at it, you see within the box here that there are these repeating sequences. So these sequences are each very close to each other. And in between these repeating sequences, there were variable sequences. And so in cartoon form, if you go from one end to the other, you see these R's, these are these repeats, and in the space between them are what are called spacers. These are these variable sequences. So just to sort of explain this acronym, more and more of these were found. They were always found in these clusters within the bacterial genomes. Regularly interspaced between these repeats were these short palindromic sequences. So repeats are also somewhat palindromic, although not always. So the CRISPR acronym actually does not describe Cas9. It describes this very strange repeating sequence here. It wasn't clear what this thing was doing until about 20 years later, when computational biologists, in particular Francisco Mojica, figured out that if you start blasting these sequences, so BLAST was a tool that wasn't around in the 80s, but as more and more genomes were sequenced and as computational tools were built, Francisco Mojica searched for matches to these spacers and found that some of them matched phages and plasmids and were perfect matches. And so this was the first suggestion that these may actually be immune systems. And then it only took 2 years after that for it to be demonstrated in labs that bacteria with CRISPR systems that had spacers that matched foreign elements were immune to those elements. So they would destroy those phages and plasmids. And this is work by my postdoctoral mentor, Luciano Marafini, who discovered that CRISPR systems actually cut DNA and that they were not working as an RNAi mechanism, which is what was the other sort of proposal. So this is what a CRISPR system looks like. So far we've just talked about the CRISPR array. These are where these molecular memories of previous phages and plasmids are stored. These are these spacers again. And here are the other elements of the CRISPR system. So there's 4 protein coding genes, one of them being Cas9 in the system we study. The other 3 genes we won't really talk about today. It turns out you need all 4 of these to make new memories, but you only need one of these genes in order to use those memories in order to destroy that target when it shows up again. And so there's also another non-coding RNA locus on the left here called the tracr-RNA. This will be very important in the work I described today. And so it turns out when Cas9 uses a guide RNA, in nature, it's actually using a dual RNA guide. One of those RNAs is the tracer and the other is from the CRISPR array. So how this works is the CRISPR array with all of these memories is made as a single precursor RNA. And then that's processed into individual guide RNAs. So these have all the information Cas9 needs to go to its target. And that's, again, these colored spacer sequences. The tracer RNA is made from 2 different promoters. So there's a long form tracr-L and a short form tracr-S. And you'll notice this black box here, it has what's called an anti-repeat. So this is a sequence that is complementary to this black sequence over here within the precursor CRISPR RNA and the CRISPR RNA. So what happens is Cas9 doesn't simply use a CRISPR RNA to find its target. It also needs a tracer RNA. Again, the repeat here and the anti-repeat on top will base pair. And that gives you this dual RNA guide. And it turns out you need the tracer as a sort of scaffold. So it holds the CRISPR RNA guide within Cas9 in the right conformation such that Cas9 can find its target. And when it finds it, it will cleave it. but it only does so if the target has what's called a PAM. So this is a sequence next to the spacer in the target DNA, in this case, which has an NGG. So Cas9 really needs two things. It needs a perfect match to its, or near perfect match to its spacer, and it needs an NGG. And this is useful because this is how the system knows not to cut its own DNA. So these guide RNAs, you might ask, are perfectly complementary to the DNA from which they were transcribed. So why doesn't it cut the CRISPR array itself? This is because adjacent to each memory, you get a repeat, and the first three bases of the repeat are not NGG. So the system will not cut itself. It will cut a phage or a plasmid as long as there's an NGG. Now, a lot of you out there are probably, if you are using Cas9 as a tool, you're most likely using a single guide RNA. This was developed by Jennifer Doudna, who recently won the Nobel Prize for many different discoveries within CRISPR biology, biochemistry, and biophysics. Her group discovered that you can truncate both of these RNAs and join them together artificially to make a single RNA, and that will work just as well as this natural guide. So as I mentioned, there's two tracer isoforms. There's a long form and a short form. Both of them have this anti-repeat, so both of them can base pair with the CRISPR RNA repeat and assume this structure, which allows Cas9 to do its thing. You can see the long form has this extra DNA up here, but both tracer RNAs allow Cas9 to cut targets. And so since the discovery of these two RNAs, it was unclear why you needed both. And because the short one seemed to do everything that was necessary, most groups have moved forward with technologies based on the short form. And the long form was mostly forgotten until we sort of stumbled into it. And I'll explain to you what it is today. So in the context of functioning as a bacterial immune system, there's two steps to CRISPR immunity. As I mentioned, when a phage infects a cell, if there's a spacer that matches that phage, the cell is immune. But if there's not a matching spacer, so here's a magenta phage, and if you look at the Rolodex of prior memories, this CRISPR system only remembers a red, orange, and yellow phage. So this cell is going to die unless it's somehow able to make a new memory against this phage. And if it does that, it will insert that memory at the leading end of the CRISPR array. So these arrays are polar such that new memories always come in at one end. So you have a new end and you have an old end. And this is actually really convenient for people who are trying to genotype bacterial strains and make phylogenies and figure out their very recent ancestral tree, because CRISPR systems can change very fast and very obviously. It's a 30 base pair mutation when a new memory is made. So a lot of strains out there may have the same spacers at the old end, but some variability at the new end. And that gives you some idea of not only where they've been, but what phages they've encountered. Okay, so step one is making a new memory. And again, step two is that whenever this phage comes back, because the CRISPR system remembers it, it will transcribe those CRISPR RNA guides, which will allow Cas9 to cut that same phage when it returns. So the cells are immune at this point. Okay, so going back to our question, we wanted to understand what regulates type 2 CRISPR systems. And to answer this question, we turn to a next generation sequencing approach. So this is where, again, core facilities have always been lifesavers for my labs, because of course we don't have our own Illumina technologies. And so what we decided to do was a TN-seq screen. So this is a genetic screen looking for factors in the host cell which either repress or activate the CRISPR-Cas system. So TN-seq stands for transposon sequencing. And so we're making mutations using a transposon. So these are mobile genetic elements that insert themselves randomly into the genome. So in this flask, I have made a library where each cell has a different transposon insertion site. Of course, some of them are the same, but the hope is that you have enough cells in there to get at least one disruption in every gene in the genome. And you can then subject that library to some sort of selective pressure. In this case, we're adding phage. And then you can ask, how does the library change after that pressure? So did some mutants become enriched and did other mutants fall out of the population. And then that tells you something about whether those genes were important for the activity. And so in this case, we're looking for modulators of CRISPR activity. And so what we did was we cloned a system where there were no memories of previous phages. So there's only a single repeat. And because of this, these cells will succumb to this phage unless they can do two things. One, make a new memory, and then two, use that memory to kill the phage. So they have to do both adaptation and interference, the two steps of CRISPR immunity. Here's an example of the type of output you might see. So an uninfected cell, that basically tells you what your library looked like to begin with. Each line here, this is where a transposon landed, and then the y-axis is how many times you sequenced that transposon insertion site. And so here you could see an essential gene in green. You're not going to get any reads in that gene. And then the rest of these genes you see, these are likely non-essential, so you do get disruptions. And that is a limitation of this technology. You can't find essential genes doing the phenotype you're interested in. But we did it anyway. And here what you see, once you add phage, this is just as an example, your insertions that were in this yellow gene disappear. So this would indicate, for instance, that the yellow gene was necessary for the activity of the CRISPR system. Because once you add phage, when you knock that gene out, you no longer see those members of the library. On the other side over here, you see these blue genes where when we disrupt these genes, the cells actually do better. Okay, so these would be potential repressors of CRISPR activity. And these are the types of things, for instance, we would be hoping to find, right? If there's a repressor like HNS that's repressing our type 2 system, that's the kind of change that we would be looking for. So when we did this screen, again, we're looking for one of the 3,000 genes in the host genome that might be repressing the system. We were very puzzled by what we got, which was that the best hits in the whole screen were actually within the CRISPR system itself. And where were they? They were actually in and around the promoter of the long form tracer. So here you can see on the top, without phage selection, we got all these different insertion sites. They're all relatively low overall because we have so many sites throughout the genome. And then when you add phage, you get this massive enrichment of cells that had a disruption in this region. And so what we did was we created a deletion of this entire region. That's called delta tracr-L. And so here you can see an immunity assay where wild type cells, when you add phage, have very low levels of immunity. Here we're basically just looking at survival when you add a phage. And when you delete this region, you see about a 3,000-fold increase in levels of immunity compared to wild type. Here in the middle, you see what this a single transposon insertion that we pulled out of the library looks like. There are intermediate increased levels of immunity. And we know now this is because a single insertion can sort of disrupt this promoter, but not completely knock it out, whereas our deletion completely removes the long form from the equation. And in blue here, this is a mutant called Hyper-Cas9. This is previously sort of the most hyperactive CRISPR immune mutant we had. And this just shows our new deletion is sort of now the new most hyper acquiring mutant out there. So this to us suggested that this long form tracer is somehow inhibiting immunity. And we can do some genetics. And so we can complement that deletion by putting the long form back on a plasmid. And here again, immune levels, which were very high, are now restored back to that wild type low level of immunity. So we wanted to know, because our screen, we asked cells to do both adaptation and interference. So make a memory and kill that phage using that memory. We wanted to know if our mutation was enhancing one or both of these activities. So we needed to somehow separate them and study them individually. So to study adaptation in the absence of interference, we knew we needed an assay where the cells would be making memories, but that there wouldn't be any phage around. So all they had to do was make a memory. They didn't have to then use it for survival. So to do this, we made cells electric competent. We then added very large amounts of a very short DNA fragment by electroporation. And there's only one NGG PAM in this sequence. And so there's only one possible spacer, one memory that can be made from this DNA that we're shoving in there. And so we can assay for whether that DNA made it into the CRISPR array by doing a PCR with one primer upstream of the CRISPR-A and a reverse primer within that predicted spacer. And so when we did this assay in wild type cells, this is sort of semi-quantitative, but in wild type cells, we didn't see any signal that these spacers were being incorporated. But when we removed that long form tracer, that inhibition was somehow relieved. Okay, so that first phase in immunity adaptation is in fact dramatically enhanced without the long form. Next, we wanted to know if the second step, if interference was also inhibited by the long form. So in this experiment, we are programming cells with a spacer. So they already have a spacer. And we're asking them, how well can you defend yourself against phage using that spacer? So we're growing cells in a bacterial lawn. So each strip here is a bacterial lawn. And the lawn is actually the space between these circles. So what the circles are is where we've dripped bacteriophage on top of that lawn, and it's tenfold dilutions. So when you dilute the phage enough, you see these single small circles, which are plaques. So this is a plaque forming unit assay. A plaque is where the phage have eaten their way through the bacteria. So if the cells don't have a spacer targeting the phage, you can see they're very sensitive to the phage, and essentially you get as many plaques as there were phage in your stock. In the second row, we've now taken wild type cells with a spacer against the phage, and you can see that the plaque forming ability of the phage is decreased by about 100-fold. And when you go in and you look at those phage that did survive this assay, you see they have mutations in the target site. So the site complementary to that spacer is mutated such that Cas9 can no longer bind. And then what we saw when we delete the long form tracer, we saw an even more impressive decrease in phage plaquing ability. And that suggests that the CRISPR system is actually more potent. So together, these two assays showed that the long form is inhibiting both steps of CRISPR immunity. So we thought this was interesting, and we wanted to understand the mechanism by which the long form was acting. So the first thing we asked was, what are the expression levels of the different parts of the CRISPR system? Is the long form somehow repressing the expression of the system? So here I'm showing you a northern blot. We're looking at the non-coding RNAs in the system, so the tracer RNAs as well as the CRISPR RNAs. In wild type cells, you can see a long form band here, but you can barely see the short form or the processed form. Tracer also gets processed. And you can't really see the CRISPR RNAs at all. When you delete the long form tracer, you see an increase in every one of these non-coding RNAs. The short form goes up, processed form goes up, and the CRISPR RNAs go up quite dramatically. And again, those are restored when we complement by adding the long form back on a plasmid. So the long form tracer RNA, which of course is absent up here in this deletion, seems to be repressing all of these other non-coding RNAs. What about the Cas operon, which is, again, all four of these genes are transcribed from a single promoter? To look at the expression of the Cas operon, we did a QRT PCR. And similar to the non-coding RNAs, Cas9 as well as two of these other Cas genes are up about 50 or 100-fold in the absence of the long form tracer. So somehow this long form tracer, when it's gone, results in increased levels of every single sort of bit of RNA in the system. So next we wanted to ask whether the long form is inhibiting the active transcription at any one of these promoters. So to ask this, we constructed some transcriptional fusions of each promoter to GFP. So we have the Cas promoter here, the CRISPR RNA promoter here, and then we did a tracer RNA reporter, which is downstream of the short promoter and the long promoter. So it's sort of right here, and it reports on the sum of those two promoters, essentially. And so for this assay, we had one plasmid that had this reporter with one of those promoters. And then we engineered cells such that they only had two parts of their CRISPR system, just the tracer RNA locus and Cas9. So these other Cas genes and the CRISPR-A are gone. And what we saw was that in the presence or absence of tracer and Cas9, these promoters of the tracer and the promoter of the CRISPR RNA are unaffected. But the promoter of the Cas operon is dramatically repressed by this locus, which again is just Cas9 and the tracer RNAs. And so now we were developing this idea that Cas9 and the long form may together be some sort of repressive complex. To ask that more directly, we purified Cas9, and we also purified the long form tracer RNA. And we asked whether it could bind to the DNA at this promoter in a mobility shift assay. So here the promoter DNA is radio-labeled, and a sort of random promoter from another GFP reporter construct was not bound by this Cas9 tracer long complex. But the Cas promoter, which we just showed is repressed in vivo, is also has a pretty high affinity. The promoter has a high affinity for the Cas9 tracer complex in vitro. And so here again, this is the minimal components of this repressor, which is just Cas9 and just the long form. And so going back to our northern blot, where I've told you that the non-coding RNAs are also downregulated in the presence of this repressor, we now know in data I won't show is that these non-coding RNAs, their levels are very dependent on the levels of Cas9, because Cas9 binds these RNAs and protects them from being degraded. So when Cas9 levels increase, even if you don't change the activity of these promoters, the levels of tracer and CRISPR RNAs also increase because of their protection by Cas9. So by repressing a single promoter, tracer long is repressing the entire system directly by repressing these Cas genes, and then indirectly by affecting the stability of these non-coding RNAs. Okay, so around this time, we made sort of a startling observation, which was that the 5' end of the long form tracer had an 11 base pair sequence, which perfectly matched a sequence within the promoter of the Cas operon. So this is that promoter that's being repressed by the long form. And there seemed to be a sequence match of 11 bases, which is not a lot, but could happen by chance. But the other interesting feature of this match was that downstream from this potential target site, there was a PAM. And again, PAM is the sequence next to the target site, which Cas9 needs to bind something. So this was starting to smell like this long form tracer was some kind of strange guide RNA, which might be bringing Cas9 to this promoter and targeting this sequence here and this PAM that was right next to it. And so we wanted to know if this was true. So what we started to do was to mutate the bases at that target site. So one by one, we introduced mutations which would break either the complementarity between the long form and that target, or that would break the PAM, which is not complementary to anything, but it's just a necessary feature for Cas9 binding. And so again, we're looking at promoter activity here using a GFP reporter in the presence or absence of this repressor complex. And so wild type cells, this promoter is repressed because Cas9 and tracer long are there. And when we start to introduce mutations, especially mutations close to the 3' end of this match, we see that that repression is somewhat relieved. And when we mutate the PAM, you get a total loss of repression. And so this is very similar to what we already know about guide RNAs, which is that without the PAM, Cas9 can't bind at all. And then with individual mutations, you start to lose binding, but in sort of incomplete fashion. So this was consistent with what we knew about guide RNAs. And I should also mention, you're probably wondering why we're not seeing cleavage at this site. What we also knew from the literature was that there's a threshold where if the guide was shorter, if the targeting site was shorter than 16 base pairs, Cas9 can bind to its target, but it can't cut. And if the sequence matches above 16 base pairs, it starts cutting. So given that we have an 11 base pair match here, this to us suggested that it could actually be sitting at the promoter and simply repressing it by preventing RNA polymerase either from initiating or elongating. Okay, so we next wanted to do the reciprocal experiment, where now we're asking not if mutations at the target site break repression, but if mutations in the repressor itself can break repression. So here's the part of that target match that's within the 5' end of tracer RNA. Again, it's 11 base pairs. And here what we're doing is introducing sort of a series of mutations, each one mutating 4 or more bases. And so we did 2 mutations here in the first half or the second half of this targeting sequence, where we're mutating each base within that clump to its complementary base. And we see, again, wild type is repressed. So without any mutations, the repressor works. Without the long form, you get high expression from this Cas operon promoter. And when you mutate this putative targeting sequence, we also lost repression. So this is consistent with the reciprocal result where you mutate the target site. What we next saw was that this DNA just to the left of that targeting site was also important for repression. So this green and blue region, when we mutated it, we again saw that repression was broken. And what we next figured out was that this DNA next to the targeting sequence looked eerily similar to the DNA in the CRISPR repeat, which was also next to a targeting sequence. So a spacer is a targeting sequence, even though it's 20 base pairs compared to 11. The adjacent DNA looked very similar, almost identical. And so this indicated that just downstream from the targeting sequence, there's a cryptic repeat, right? So tracer, we know, has an anti-repeat, which binds to the CRISPR RNA repeat. And that's shown here. This is that binding, which gives you that dual RNA guide. We know that both the long and the short forms have that anti-repeat, which allows them to bind the repeat and form these structures. And what we're now saying is that the long form has its own cryptic repeat, which sort of mimics the CRISPR RNA repeat. And this gives the long form RNA this opportunity for its repeat to bind to the anti-repeat. So here on the right is the anti-repeat, and here it's binding to its own cryptic repeat at the 5' end instead of the repeat from the CRISPR RNA. And so now we were thinking, okay, so this long form tracer is actually forming a natural single guide RNA. So, while we invented a single guide RNA within the last 5 and 10 years, which has been tremendously useful, it turns out nature had actually been using a single guide RNA for a very long time beforehand. And the structure of this single guide looked very similar to the single guides we use, with sort of 2 exceptions. Again, one, this targeting sequence is 11 base pairs instead of 20. This is what allows it to repress instead of cleave. And there's also this very conspicuous upper stem extension, which is absent in the single guide, which is a more simple fusion sort of in this region. And so what we next wanted to understand was if this structure was actually real. Is this actually forming according to our model? So we went back to our GFP promoter reporter, and we asked, okay, what happens next if we mutate these regions in the 3' end of tracer, which we know are essential for being, the scaffold that normally binds the CRISPR RNA and holds it with Cas9. In our model of how the long form folds, it's still acting as a scaffold. It's just binding to itself instead of a CRISPR RNA. And so in fact, when we mutate the bulge here, or the upper and lower stem, or what's called the nexus here, all of these mutations broke repression, consistent with this thing behaving as a single guide. When we mutated the upper stem extension, the repressor still worked. And actually, I won't show it here, but we deleted the whole thing and the repressor still worked. So we really have no idea what this thing does. We think it might be involved in de-repression, which is something we're very interested to explore more, but at least in terms of repression, it doesn't seem to be playing a role. So next, to really rigorously test this model where this is the structure that's forming in the long form tracer, we started making mutations in the lower stem, which we knew from that previous analysis would break repression. So if you mutate the left part of the lower stem, you no longer have the ability to form the base pairing here, and you break repression, shown here. Same thing if you mutate the right side, you break repression. But if you mutate them both simultaneously to their complement, you actually restore repression, shown here, suggesting that this lower stem is likely forming in the long form tracer. And this, it doesn't restore the function of the repressor to wild type levels, but if you look at another mutant we made where we're completely changing all of these bases, but now introducing some more GC base pairs, maybe to make this more stable, you see that this V7 mutant almost completely represses as well as wild type. And this to us is really interesting, actually, that Cas9, it needs some of these structural elements to bind these RNAs, but it turns out the identity of these bases is not super important. You simply need a lower and upper stem and a bulge here. But the sequence is somewhat flexible, and we think that's another really interesting feature of Cas9. And it might suggest that in nature, Cas9 can bind to more guide RNAs than we think, right? If RNAs by chance have a similar structure, they might get bound. And that's something we're exploring as well. Okay, so if this long form tracer RNA is indeed, this single guide, then we wanted to know if we could reprogram it. So can we change that 5' end, that 11 base pair sequence, which specifies a target, and can we now assign Cas9 to repress a new promoter? So we changed the sequence here now to a sequence that targets Pbla-Z, which is another reporter promoter we had lying around in the lab. We show that compared to the wild type targeting sequence, which targets the Cas gene promoter, this reprogrammed sequence, if it's 11 base pairs, gives you about a log reduction in GFP expression. So it can be reprogrammed. We think this difference is not as nice as what we see for the Cas operon because this target is actually a bit downstream from the transcriptional start site. And we think the closer you get to the start site, and this has been shown in other work, the better repression you get. We also show that if you increase the targeting site to a 15 base pair match, you can see even tighter repression. And so this is also interesting because nature has evolved for whatever reason an 11 base pair repressor. And this might be so that the system is not completely turned off. So we do still see leaky expression despite this repression. We do still see some basal immunity against phage. So having a repressor that's not a complete off switch may allow the system to have some immunity while preventing some autoimmunity. And then, of course, if we increase the match to now 17 or 19 base pairs, that's above the 16 base pair threshold where now Cas9 becomes a nuclease. And so here's an in vitro assay where we're now doing a cleavage assay. And compared to the canonical single guide, which most people use, which has a 20 base pair match, the 19 base pair reprogrammed long tracer essentially behaves in an identical fashion and can cut quite well. Okay, so why, again, would a CRISPR system have such a strong dimmer switch when we've shown that when you remove the long form, you get 3,000-fold more immunity against phage? That seems like a good thing that cells might want to have all the time. Well, the flip side of that, as I've mentioned, is that there's increased autoimmunity. So in this assay, we're mixing wild type cells with those that lack the long form. So you start with a 1-to-1 mix and you sort of compete them against each other over time with serial dilutions. And after just 3 days, we basically no longer could detect the mutant cells, which didn't have the long form. So we're not adding phage in this assay. We're simply asking them to grow in media, but in the absence of phage, wild type cells have a very strong advantage. And I think this demonstrates that balance between immunity and autoimmunity that the cells in nature likely have to navigate. So looking back now at what we think is happening, as I mentioned, there is an intrinsic regulator built within this CRISPR system, which is composed of the Cas9 protein, as well as this long form tracer. And that tracer targets the promoter of the Cas operon and represses it, and in turn represses also these other non-coding RNA loci. And we're starting to sort of think of this as a genetic circuit where we know there's competition within Cas9 for the long and short form tracer RNAs. And depending on whether it binds long or short form, it might repress itself better or worse. So you can imagine factors affecting either the stability or transcription of these two forms could affect how active the system is. Another potential site for regulatory input is the CRISPR RNA. So if, for instance, transcription increased of the CRISPR RNA, we know CRISPR RNAs can bind to the long form tracer. And when they do so, Cas9 is, in this case, acting as a sort of normal immune effector. It's going to use that CRISPR RNA to find a phage target. But this conformation of the long form tracer is mutually exclusive with its conformation as a single guide repressor. It cannot accommodate both its own 5' end as well as a CRISPR RNA. And so anything that affects the levels of the CRISPR RNA will also affect how well this repressor is working. And of course, there's other factors yet to be identified that might, for instance, um, target this upper stem extension. So small molecules, RNAs, proteins that bind to this region and maybe inactivate it could also inactivate the repressor. And also anything that inhibits Cas9 would also inhibit repression and lead to potentially a burst in Cas9 expression. So we're exploring how all of these things play out in live cells with emphasis on this question of how these systems get turned on when needed, for instance, before or during a phage infection. And the other question is, what kind of new tools can we make based on these observations? So as I mentioned, if we find out a use for this upper stem extension, maybe it's involved in inactivating this repressor, you could imagine making some really interesting new guides that are now regulatable guides. So if we knew what that small molecule was, we could now turn on and off guide RNAs while we're editing something or while we're repressing something. You can also imagine multiplexing and giving cells several different types of guides. So in the same cell, you could give it a canonical guide to do some editing and a non-canonical guide to do some repressing. And we know there's a lot of hard to edit cells out there. Why are they hard to edit? In some cases, it's because they're expressing repair genes, which are preventing the type of repair that you desire. So if you want non-homologous end joining, but you're getting homologous recombination, can you transcriptionally repress the HR genes with a tracer long analog while editing the gene you want to edit with another guide RNA? So we think there's a lot of cool things you can do with these sort of new types of guides. So with that, I'd like to thank the folks who did the work on this paper. Rachel Workman, a graduate student in my lab, and Teja Pamri are the primary authors on this paper. Teja is a technician. Marie and Bin are also authors on this paper. So this was a real group effort. And it was incredible just as the lab started in the first couple of years to amass such a great team of really smart people and have them all working together on the same thing. It was a really cool experience. Of course, I'd like to thank the GRCF core. They've done sequencing for us on this paper and on other projects we have going on in the lab. And it's a real pleasure to have them on campus and of course, to be giving this seminar. We appreciate you. And I'd also like to thank my home department of molecular biology and genetics for the startup funding that funded this project. So that is all I've got.